Hello and welcome. Today, we're diving into the incredible world of gene therapy, moving from lab concepts to real-world treatments that are changing lives. This is translational gene therapy in action. We're seeing amazing breakthroughs. Imagine correcting genetic diseases with a single treatment. Luxturna is restoring sight to people with a specific inherited blindness. Zolgensma offers hope for infants with spinal muscular atrophy. Hemophilia B patients can now produce their own clotting factors thanks to therapies like hemgenics. And in a major milestone, CRISPR gene editing is being used in approved therapies like Casgevy and Lyfgenia to tackle sickle cell disease and beta thalassemia by fixing the patient's own cells outside the body. Even in cancer, CAR-T therapies, which genetically engineer a patient's immune cells, are fighting certain leukemias and lymphomas, and newer approaches are targeting solid tumors. Rare metabolic disorders are also seeing progress, with treatments like Kebilidi delivered directly to the brain. These aren't just theoretical anymore. They are approved therapies making a real difference. But how do we get these genetic fixes into the right cells? That's the multi-billion-dollar question. Safely and effectively delivering the therapeutic gene is the biggest hurdle. This is where sophisticated engineering comes in. For a long time, viruses, carefully modified to be safe, have been the go-to delivery vehicles, or vectors. Adeno-associated viruses, or AAVs, are popular because they're generally safe and can target different tissues. Scientists are constantly tweaking the AAV's outer shell, the capsid, to make it better at hitting specific cells. Think of it like programming a GPS for the gene delivery. They're also working to make these vectors less likely to trigger an immune response and easier to manufacture at scale. Lentiviruses are another type, often used when modifying cells outside the body, like for CAR-T or the sickle cell treatments, because they integrate the gene into the cell's DNA for long-term effect. Safety engineering here is key to prevent unwanted genetic changes. While viral vectors are effective, they have limitations like potential immune reactions and size limits for the genetic cargo. So researchers are developing non-viral alternatives. Lipid nanoparticles, or LNPs, tiny fat bubbles, showed their potential delivering COVID-19 mRNA vaccines and are now being adapted for gene therapy. They're potentially less immunogenic and easier to produce. Polymeric nanoparticles and even physical methods like electroporation, which uses electrical pulses to open temporary holes in cells, are also being explored. Non-viral methods offer safety advantages, but getting them to be as efficient as viruses inside the body is still an ongoing challenge. And what about gene editing tools like CRISPR? Delivering the components needed to precisely edit DNA also requires these delivery systems, whether viral, non-viral, or physical methods. Despite the successes, challenges remain. We need to improve delivery efficiency, manage immune responses, ensure long-term safety, make these complex treatments affordable and accessible, and guarantee the effects last. Much research, supported by organizations like the NIH, focuses squarely on refining these delivery technologies. Improving how we deliver these genetic instructions is key to unlocking the full potential of gene therapy for many more diseases. The future looks promising as we continue to engineer better solutions. Thanks for tuning in.